My name is Higgs and this is my friend Boson. Welcome to the Higgs world.
Thanks for the introduction, Higgs. In this presentation, our goal is to offer you a quick summary of the research and conclusions. We follow inquiry-based learning model where students take lead in learning.
First, they get a quick review of the research via the videos. Second, they review the paper to learn and understand key concepts. Finally, they can follow up with further research on their own to dig deeper. In the end, students would have learned something new, latest and relevant. Isn't that wonderful, Boson?
Great point, Higgs. Please follow the links to review the research in further details. We want to thank all the authors and publishers for making this research public, thereby advancing science and innovation. Please like, share and subscribe.
Today, we are going to review a paper titled, Creation of a Prototype Biomimetic Fish, to better understand impact trauma, caused by hydropower turbine blade strikes. Fishes and marine mammals are at the center of many studies because of their unique adaptations to sense the environment and move efficiently to obtain resources while evading potential threats. Biomimetic model organisms could be useful surrogates for live animals in many applications, if the models have sufficient biofidelity.
Wonderful, Higgs. One such application is for use in field and laboratory tests of fish mortality, associated with passage through hydropower turbines. Dose-response relationships have been created from these data, but the exact relationship between fish mortality and the actual forces enacted on fish during simulated blade strike testing remains unknown.
That is a great point, Boson. This research describes methods used to create a prototype biomimetic model fish, composed of ballistic gelatin, and covered with a surrogate skin to better approximate the biomechanical properties of a fish body.
That is interesting, Higgs. Frozen fish were scanned with high-fidelity laser scanners, and a 3D printed, reusable mold was created from which to cast a gelatin model. CAD models for use in additive manufacturing of molds. The resulting model had a statistically indistinguishable tissue durometer, to that of real fish tissue. Preliminary blade strike impact testing suggested its overall flexibility was similar to that of live fish.
I have more to share, Boson. Gelfish was designed with biofidelity as its guiding principle and the results suggest initial experimentation was successful. Study suggests that the use of ballistic gelatin and 3D scanning to produce reusable molds worked well to recreate the overall shape and basic biomechanical properties of a real fish.
Wow, that is amazing, Higgs. Ballistic gelatin was easy to work with and could be modified to account for small changes in tissue firmness related to different species. The response of the Gelfish model from simulated impact conditions suggests it may be slightly more flexible than real fish, but more tests are required to quantitatively confirm its biomechanical properties.
The paper suggests that the process used to create models needs further development to augment its biofidelity and make it more versatile for use in other applications, including irrigation and water pumping stations analysis.